Very good afternoon to everybody. I am privileged that I have been given this opportunity to talk to some learned people across the world. This is a very good initiative to bring people together on what's happening on the research development and the engaging farmers, people, the seed, systems in case of beans. So I am Parvez Ahmed Sofi. You can call me Parvez, or you can call me Sophie, the way you like. Yeah, I am working currently as a professor of plant breeding, and I am the department chair in the Kashmir University of Agricultural Sciences and Technology. It is one of the universities in the Indian state of Jammu and Kashmir. So in India, there is a national agriculture research system where we have around 78 agriculture universities, and ours is one of them. We are actually located, as you can see from the picture, that our campus is also in the lap of mountains. So this is a typical Himalayan region with mountains, everywhere. And maybe most of you know that our place, the Kashmir, is called as paradise on earth. I'm not sure whether anybody has visited this place or not, because in the past, when there were some issues with the security and other things, there was some problem, but that's now over, and life is absolutely normal now, everything okay. So I was given an opportunity to talk to you people on how we do about developing high yielding, nutrient dense and resilient varieties of beans in the Kashmir Valley, because it's as I told you, it's a mountainous area, and it had its own typical problems and bottlenecks. The major of problems being it's a fragile area, it's vulnerable. We are on a very kind of negative side of the Richter scale, where we are prone to earthquakes and other things. And we have very small holder farmers, marginal lands, hilly areas. We are sometimes inaccessible when the road gets blocked from rest of the country. And amid these challenges, we have held ourselves on, and we have demonstrated our resilience as a very resilient people who have gone through many problems, but still managed to be one of the best places in the world, and one of the best in education, I must say, with all the pride that I have. Let me introduce ourselves. That, as I told, we are one of the states of India. Around 30 states there are, and we are one of them. And in that state of Jammu and Kashmir, there's a place, Srinagar, where our headquarters is. You can see the beautiful gate of the university. Background, you can have very good clouds and trees. Actually a wonderful place, campus, one would love to be in and visit, sometimes. So as an institution, our distinctions are that, as I told you, that we are one of the resilient, we have demonstrated our resilience despite all our challenges. And currently we are the fifth best state agriculture university in India. We have a system of accreditation by Indian Council of Agricultural Research, and we are graded A. We are the first among state agriculture universities in innovations, uh, among all these universities. We are the first university that adopted the startup and innovation policy to promote entrepreneurs among faculty as well as students. We have a technology business incubation center where we train students and faculty for converting their innovation ideas into startups and companies. In India, we have a system by Department of Biotechnology where they give very good grants for innovative proposals that can develop solutions for the problems that are faced on different social fronts, including agriculture. So we are the only university in India that has got 15 such grants. And during the last few years, we have, as I was telling, we have focused on innovation, startups, and translational research, and we have been able to get 92 patents for the innovations that we have developed. And during the same period of time, we have got 72 startups established by the students and faculty. And out of those 72 startups, 25 companies have, been established who are currently running their business. As an institution which is involved in research, of crops, livestock, forestry, fisheries, sericulture, engineering, basic sciences, community sciences. So we are interested in, kind of transforming agriculture in the state by giving technology backup. And one of the technologies that we develop is the improved varieties of crops, including beans. And during these 40 years of our existence, we have developed around 124 crop varieties, including almost 20 in legumes, including 5 in beans. So some of our distinctions, kind of translational research is that we are the first in the world to clone a goat that is world's prized goat that's called as pashmina. So we have been able to clone that goat. So in India, we are the first university to have edited sheep through gene editing technology. It's very difficult in animals, but we have done it. We are also developing engineered vaccines for poultry and livestock, causing so many diseases, those bacteria and other pathogens. And we are also working on regenerative stem cell technology for healing wounds and other bruises in animals, including humans. And we are in the way to have an international patent for that technology. So this is something I was trying to introduce ourselves, who we are, what we are doing, and where we stand in terms of our scientific endeavors, and in the country where we are in different indicators of scientific knowledge development and progress. So coming to the today's topic, that is about beans. As far as legumes are concerned, we know that all the pulses, they belong to the legume families. And in our state, we have many legume crops, but most of these come under the underutilized crop category. So we have beans, we have cowpea, we have chickpeas, we have lentils, we have mung beans, and we have the field peas. So all these together, constitute the legume, cropping system in our state. Even though there are some minor legumes like faba bean is grown somewhere, likewise there is that black gram being grown somewhere. Many other legumes are being grown at many places on a very small scale, especially in kitchen gardens and other places, but not on a commercial scale. In beans also, we know that there are many cultivated species. Almost 80%, 5% area in the world also is, covered by Phaseolus vulgaris, or the beans that we are talking about today. And we also have some, in pockets, we they grow that scarlet bean, but it's very, very small scale, almost negligible, because of some obvious reasons. Because its seed is not that tasty, its pods are leathery, the plant type is not good. It is a, and the tendril, vine which goes very up, to larger heights. It is sometimes late in maturity, so people don't grow it that much. So beans are the most important pulse crop in our state. It is in fact globally recognized, for its taste and organoleptic qualities. And we are going to get geographical indication for our beans. I think the process is going on. Some paperwork needed to be done, and we are almost at it. So this is the typical farming system of beans. If I can show through some pictures, you will not see big commercial farms of beans in our state, but it will be on, as I told you, it is a hilly area. People will use whatever land they have left after they cultivate rice. Rice is the major field crop in our state. And after that, whatever land is left, some of it goes into maize, some of it goes into different pulses, including beans. And one of the patterns of cropping in our state for beans is that it is intercropped with many, many different, these crops like maize, like cowpea, like tomato, potato, mixed, cropping systems are there, especially in far-flung areas where people have a highly diverse, diet. In fact, you can see in one of the pictures on the top left, there is a big, row of the scale in between, the bean, kind of plot, and there are many other crops growing on the sides like maize and other things. So it's a composite kind of block. Whenever you go to bean fields, you will not see those big, fields of beans. But it's a very, very important crop, especially in small and marginal farming systems, where it not only, takes care of the nutritional security because animal meat or protein is very costly, but it also fixes nitrogen. That way, it contributes to ecosystem sustainability, and it also fetches a very good price in the market nationally and also internationally, because there are so many people of Kashmiri origin who are living elsewhere in the world. They have a great demand for this the local beans. They call it rajma in local language. So in fact, it's a very, very prized crop, as far as I know. It fetches very good price. That's the only kind of motivation for farmers to grow it. Otherwise, in terms of its yield, its productivity, and its resilience to disease and other things, it's not as competitive as rice for the farmers to grow. So if we look at the bean market classes, in our place we have the dry beans that we take after drying the seeds. We also have green beans, which we call as French beans, which are taken as vegetables along with the seed and pod. And there is also one class of variety, we call them as shell beans, in which that when the beans are almost physiologically mature, but the pods are not dry. Only the seeds are taken after cooking, but the pods are not, because pods have somehow become a little bit leathery and they are not good for eating. So these are the three different classes of beans that are grown in our place. There is one more class that is not present in our place, that is that popping bean. So it is in some areas in the world an old land race called as Nuna, which is being grown as a popping bean. But in our place, it's not there. So we are one of the very good hotspots of diversity as far as beans are concerned. Because Kashmir is a place that has been visited by so many people. During India's rule by British people, during the Mughal invasion, people have come from Middle East. People have come from many other places, and when they have come to Kashmir, they have brought samples of seeds with them. And because of the trade relations with other countries, so this the germplasm has been kind of transported here and there. And our place, they have got adapted and got acclimatized. And today we are seeing our place as one of the very good hotspots of diversity in terms of seed color, seed shapes, the length, breadth, the type of seed, the maturity classes, the plant type, the quality, everything. If you look at, we have a very highly diverse germplasm, mostly in the form of land races that farmers grow with them. Because in legumes at our place, despite high yielding varieties have been developed and released, but farmers somehow have a kind of that love and belonging for their own land races, because those land races have adaptability and they have quality and they are somehow a part of their living culture. That's why they continue to grow those land races. Similarly, you can see a lot of diversity in the pod shape, pod color and length. So many colors and types are available at our place. So as far as beans are concerned, from a research point of view, we have so many challenges that we are going to address, trying to address at our institutional level in collaboration with farmers, the departments of agriculture, the industry, and many, policy, making bodies from the government. So some of the challenges that I can pick up for your information is that, as I told you, ours is a small and marginal farming system across all crops. So they are mostly grown as low input farming systems. You will not find much of nitrogen and phosphorus being given. Mostly it is rain-fed pulses, maize. Only rice in our place has an irrigated ecology. Rest of the crops are mostly grown as rain-fed crops. So another problem is that is happening in places like Kashmir. As I told you, we are a very vulnerable and fragile area, and this climate change is more than obvious in our place as compared to other places. I'll tell you because in India, other places the temperature is already 40 degrees centigrade, and if it goes to 41, it doesn't make any difference in terms of perception of climate change. But if we come to our place, where the temperatures used to be maximum 32 degrees in, kind of peak summer, now they have reached almost 37, degrees. This year we had a very severe drought and heat stress to our crops. So they are becoming a major challenge again, for our bean farming systems. And what happens that the yields gets reduced and they again become less competitive and they are relegated to much harsher environments. Farmers would keep their prime lands for rice and other crops where they expect assured yield because of the assured irrigation. Then adding to the problem are so many diseases like being common mosaic virus, the anthracnose, angular leaf spot, and the fusarium wilt that is again reducing the yields. And beans, I must say, for the information of those who are attending this meeting, pulses in general are low yielding as compared to cereals. The reason being that in pulses, you can see that they are a protein rich crop. And for making 1 unit of protein, the energy required is more than what it is required for making 1 unit of carbohydrate for crops like rice and wheat. And some of the energy in the legume crops is going to nitrogen fixation because making nitrogen from atmospheric nitrogen is an energy intensive process. You can well assume this situation from the fact that the commercial process of making nitrogen fertilizers like your urea that is produced by the Haber-Bosch process, it is one of the most energy consuming process or reaction in the world. So you can well assume that how plants might be, kind of accounting for that kind of energy. So when the energy is lost in the process development and process sustainability, yield is going to be, reduced. That's why pulses, legumes are in general low yielding as compared to pulses, and they are less competitive in that way for the farmers to be grown as major crops in any farming system. Even if you look at the country level, while as rice in India has more than 100 million hectares, I suppose, and wheat and other crops, you will see pulses not more than say 20 million hectares or something like that. Put all the pulses together. So that is the reason. One of the reasons is that pulses in general are less competitive as compared to cereal crops. So when we started our research, into pulses, one of the first things that we did is that we tried to see that what's the origin of these beans in Kashmir. And because in beans there are two major gene pools. One is the Andean mountains, and another is the Mesoamerican gene pool that's in the mid of America where they have originated. So from those two places they have gone to whole places across the world, and also in our place they have been they have come and they have adapted, and from both the places of their origin, both Andean as well as Mesoamerican. So a very small way to identify Andean and Mesoamerican is seed size. So the beans with bigger seed size are the Andean beans, and the beans with the Mesoamerican are they're very small in size. So at the biochemical and molecular level, there are two markers. There's a fasolin, marker gene which is used for differentiating these, Andean and Mesoamerican. So this there are two types of fasolins. One is T, another is S. T, stands for Andean, and S stands for Mesoamerican. So it's very easy, to see that what are the changes in the ITS markers flanking those fasolin genes. and we can identify the material, whether it is Andean or whether it is Mesoamerican. So we tried to see, and we found that our material was having both Andean and Mesoamerican origin, as well as there were some mixtures also, because the germplasm has is being transferred to all the places freely now and for some time, and it has come through travelers, it has come through many different sources. In that way, that that mixture has happened. So then we started our breeding program with extensive evaluation of different germ plows from our own land races, from the National Gene Bank, and from international gene banks, especially the gene bank that is in Colombia, the CIAT, what they call as International Center for Tropical Agriculture. And there is also one gene bank in Germany, which is called as IPK, Leibniz Institute. And there, from we also got some material. And then we evaluated it for its suitability, under our Kashmir conditions. Many of the lines did not flower because they had come from some tropical regions, and we are a purely temperate ecology. So they could not flower because of less heat and those heat hours that they could not meet out at our place. So after screening, we came in, with a core set of lines which were diverse in different, traits, and they were representing most of the variability in different traits like seed size, seed color, seed shape, and plant height, different, agromorphological traits. And based on those, we generated a core set, which we analyzed through then markers, also using both SNP and SSR markers, both types of markers, random as well as linked markers. And then we tried to develop high yielding varieties that could go into the farmer's field. And because of our efforts, we were able to identify many high yielding varieties. You can see these producing very, very good yield, at the farmer's field as well as our research station. And as a result, during the last 10, 15 years, we were able to, release around 5, 6 varieties of beans, which have been in the farmers, fields and the seed systems, and have helped farmers to get higher returns, in terms of better production and price. So then we also try to see that what is the allelic diversity and what is happening at the gene level in terms of the yield and traits, that does our material, have some variation. We could see that, and there was more of kind of differences in those exotic lines as compared to the local lines, because maybe those exotic lines were coming from many different places, and our local material was coming from a very small, geographical area. That's why it had been under almost similar conditions growing, and the same evolutionary pressure it was undergoing. And while as the material coming from exotic places might have evolved under diverse ecological conditions that may result in greater heterozygosity, greater allelic diversity in exotic material. Then our second focus area, as I told, is that we are confronted with the big issue of climate change, and climate change at our place is much more pronounced as compared to other places. In fact, it's a globally accepted fact that small and marginal farming systems are more vulnerable to climate change as compared to big farming systems like the ones which are in United States or other Western areas. So what we did is we tried to see that how could we target this trait and what trait we should use to identify drought resilient and heat resilient lines. So we had more of our focus on root traits, because we know that roots are the trait, organs which are actually helping a plant to battle it out. Because once there is drought, there is definitely shortage of moisture, and it is only the roots who can help plant to acquire moisture from different soil layers. So we started our breeding program by extensive phenotyping of our material for variability for root traits, and we could find two types of roots: ones which go down and have better root distribution at different layers, especially at the bottom of the root, and the roots which were having a very, very different structure where they had some roots at the top, but at the bottom they are having very little distribution of roots, and it doesn't allow a plant to kind of absorb those water and nutrients from the lower parts of the soil. Because what happens under drought and heat stress, much of the water that is available in the upper portion of the soil gets evaporated also, and then add to it that evaporate to try this transpirational demand of the plants under high temperature and drought. So that depletes the soil moisture at the top, layers, and the moisture is mostly at the bottom of the soil. So you need plants that are able to go deeper and have more biomass allocated at deeper layers so that they can fetch water and make plants survive under such testing conditions. So we started our program, with the extensive phenotyping for roots, and then the lines that we found better were taken to field testing for different traits, and then they were, kind of taken forward in the breeding program. So what we found in our root research is that there was different plasticity of genotypes to the drought stress. Some of the plants maintained almost comparable root lengths. Some of the plants had better root, lengths under these, drought conditions, while as some of the plants, had, severe reduction in both shoot and root traits under these drought and heat stress conditions. You can see that how beautiful correspondence we were able to find between the top of the plant and the bottom of the plant. That's why it's called as the hidden half of the plant, because we cannot see roots from, the top of the soil. So we have to dig deeper to see that what happens. So in order to be able to study roots, we do not do these experiments in the field, but we do it in the, greenhouses where control conditions could be created, and where, plants are given almost uniform conditions except the drought. And then, the medium is chosen in such a way that we are able to harvest the roots reliably, and we are not losing the roots, so that we could actually see the genetic potential of different lines under drought stress. And this, phenotyping, can be taken forward by using new machines, new softwares, and new phenotyping methods. And one of the methods is what is called as root scanning, whereby you can get in-depth information in the about the distribution of roots, that is there, in the soil, happening because of this, the change in the plant system once it encounters drought. And the beautiful part of it is that what you cannot see from the gross structure of roots, even if you are able to get the roots from soil or the growing medium, you are not able to tell what is actually happening inside the root. But when you scan them using a high resolution scanner, flatbed scanners, you can see the differences becoming quite obvious. You can see two different plant types having two different distributions of roots. One is having a sparse, these fine root, system, and another genotype having a very dense fine root system. So that those fine roots allow the plants to extract more moisture from soil and take it to the top of the plant so that plant could survive under those drought conditions. And in addition to that, we also try to see what is happening at the top, because plant has to fight at two levels. One, it has to acquire resources from the soil, and then it has to be able to conservatively use them so that the period of drought or the shortage of water is being taken care of, and plant doesn't wither away or it doesn't senesce early so that the yield is lost. So we use different, phenotyping protocols, like the canopy temperature we use as an indicator of drought stress. We use, some, membrane damage potential or the oxidative, stress that is happening in the plant under, drought stress. And we also go for some imaging, techniques using NIR cameras, near infrared cameras, to see, to check the health of the chlorophyll that is in the plant. Because once the chlorophyll gets damaged, there is no photosynthesis, and once there is no photosynthesis, plant can neither, progress its shoot, nor it can give biomass to the roots so that roots could go deeper and fetch moisture for the plant. So these things we are doing, and we're trying to develop an integrated protocol. We have, published, many papers on that, how to integrate root and shoot systems so that we could, effectively develop the resilient plant varieties. Then we also try to see what are the genes and proteins that are, kind of, contributing to them. So we, kind of tested our two contrasting genotypes, one which didn't go undergo changes in the drought system and in fact had better root system at the deeper layers, and the one which underwent severe changes under drought stress. So we could find that there were so many proteins and metabolites that were kind of differentially expressed, in these two contrasting genotypes. One the drought tolerant, another the drought susceptible, and most of those proteins were related to stress tolerance and biosynthetic processes, because once those processes get disturbed, the whole biosynthetic pathways get blocked, and then the plant is not able to produce biomass. Then we also had one more issue, that is the phosphorus, which is becoming very, very important in terms of soil health, because phosphorus has a problem that it is not available readily. Once we apply phosphorus, it gets fixed in the soil and it's not readily available. And when there is a stress, there is problems in the photosynthetic pathway, there is problem in the biochemical pathway, and phosphorus itself is a part of DNA. So when the phosphorus becomes limiting, the DNA process becomes affected, and that creates problems in cell division, DNA synthesis, and eventually the plant performance gets impacted. So what we did is that we tried to see that what's the effect of phosphorus starvation, as well as the effect of phosphorus solubilizing bacteria that are there in the soil that help a plant to solubilize phosphorus, because as I told you, phosphorus is not readily available. So plants needs to get it solubilized so that it could take up. Within the plant it is mobile, but outside the plant it is highly immobile. And I was told by one expert that in one day a phosphorus molecule can just move 2 millimeters. That is the level of immobility of this phosphorus nutrient. And we could again see that roots here also play a great role. You can see how phosphorus solubilizing bacteria create a very good root system, especially at the top of the soil, because phosphorus is not mobile. It is at the top of the soil. It's not at the bottom like water and nitrogen, which are highly mobile, and they go down the soil, and plant has to have a deeper root system to kind of forage those nutrients. So we are using this. We have not published it yet, and because we are carrying this forward to see that what are the underlying mechanisms into this kind of plasticity of different genotypes. We also tried to use different omics approaches, because this is the age of omics. There is genomics, there is proteomics, there is a kind of transcriptomics, there is many omics, metabolomics, ionomics, interactomics. So many omics studies are nowadays being taken up for understanding the molecular mechanisms of plant responses to different environmental conditions. And we could see that under phosphorus starvation, so many biochemical pathways, especially the amino acids, were being kind of derailed, and even the plant structure in roots and shoots was getting changed, because that was a kind of adaptive mechanism. Plant tries to adapt itself to kind of protect itself from the damage caused by phosphorus starvation. So those kinds of changes we were able to see and report. This has recently come in plant science. Then another problem that I told is the bean common mosaic virus. So you can see what happens to the leaves. It totally kind of destroys the chlorophyll molecules, and then plant is not able to photosynthesize. Especially if the disease comes before flowering, you will not harvest anything, be sure about it. But if it comes after flowering, and at that point of time, since there is no need to produce photosynthetes, in fact the focus of the plant is to translocate the photosynthetes. So at that point of time it may not cause that much of decline in the yield. But if it comes at before the reproductive stage, at the early vegetative stage, you will not harvest anything from the field. So we were able to screen a huge set of lines for this disease, and then we were able to zero in on some candidate lines, and we could find that one of the lines contained a dominant I gene, which confers resistance to bean common mosaic virus. You can see the response of this line SR3 under the inoculation conditions. You can see it in the greenhouse, how it maintains lush green growth despite inoculation. And then you can see when we take it to the field for larger scale evaluation, it has a very good performance in the field also. And based on its this yield, based on its resilience, this variety was released in 2022 as a state released variety, and now it is in the seed chain, and it is also being used as a national check in the all India bean improvement program. Another disease that is very problematic in our conditions is the angular leaf spot. This disease not only kind of kind of leads to the decline in the plant growth, but it also causes quality issues because the pods get deformed, the seed gets deformed, and it doesn't have any market value. So one of our PhD students worked on it. She used a 200 genotype set for SSR and SNP genotyping and the field phenotyping for 3-4 years, and then she was able to identify certain lines which had high amount of resistance to this dreaded disease. And then she was also able to map some QTLs that could kind of be controlling this resistance to the disease. And we were able also to validate certain markers that were already reported to be linked to this resistance genes that give resistance to angular leaf spot. And we were able to validate the results which are reported by many earlier workers. And this work is also under process of being compiled into a publication, and we are at it. Then we also try to see what happens at the biochemical levels. In fact, we are doing it for all the disease like BCMV. We are currently doing it, in alternate this anthracnose. We are doing it, and in this kind of common bean, in this Pseudocercophora pathosystem, we are trying to see what is happening at the biochemical level under angular leaf spot pressure. And we could see that there is a lot of role for different protective those enzymatic and non-enzymatic components like the proline, the catalase, the MDA, the phenols, flavonoids, all those antioxidants that help a plant to overcome stressful conditions. Again, I told you that for this anthracnose also we use the same strategy that first we went for the large scale characterization of material, and then we zeroed in on some lines or some 188 lines. Then we did a genome-wide association studies using 5 dreaded races of this anthracnose, and we were able to identify this SR3 line, which I showed that it is a multiple disease resistant line. It was having resistance against 4 of the 5 races in that material, and also one more line WB966 that was also, and it is being used as a parent now in the hybridization program to develop varieties. That's how we did it. We tried to see the genetics of this resistance also, and we could find that it is governed by a single dominant gene when we made a cross between this resistant line and the susceptible line. Then what we did is we tried to see that are there any lines which are having resistance to multiple diseases that come in the field, because most of these pathogens are there in the field in terms of different forms of inoculum. So one of my PhD students worked on this, and she could identify 5 lines which were having resistance to all the 3 diseases, that is BCMV, the angular leaf spot, and this anthracnose. And this was published this year only in Genetic Resources and Crop Evolution. Then another problem that comes in our place, and it is becoming more obvious in the high temperatures, that is the shattering of pods. Shattering is a domestication related trait. The wild pods shatter because of seed distribution dispersal imperative, but under cultivated conditions it's an undesirable trait, because if the pods shatter in the field before harvest, farmers are not going to harvest anything. So that to tackle this problem, we again used the physical and biochemical traits to identify certain lines, and we were able to identify 2 candidate lines for this pod shattering resistance. You can see those lines, they do not even shatter by mechanical breaking. The pods will break, but the seeds will not come out. Whereas your susceptible varieties, the moment you crush them in the hand, and all the seeds will fall down. We were able to identify the role of lignin layer that imparts strength to the pods, and how pods can be differentiated very early in the selection process using this important biochemical marker. As I was telling you that Kashmir is known for the quality of its beans. We have this pride of saying that our beans are globally recognized as one of the tastiest and organoleptically very superior beans. So we try to see that what is the mechanism behind this cooking quality, and what makes the beans soft. We published in 2-3 years before, one of my master's students worked on this trait, and we could see so many physical traits that could be used to kind of assess this. And we develop a very simple test, low cost test using simple alkali potassium hydroxide. The way we do it in rice, we standardize this test for beans also, to see that which beans are going to be easy cooking and which are going to be hard to cook. What we see that in this potassium hydroxide test, if the beans disperse quickly after 24 hours, and then what happens that those beans are going to be easy to cook. Otherwise, if they remain intact, they are going to be hard to cook. We also try to see the water absorption as an important trait, simply because cooking is a very tedious process and it could have relative differences between two different people. The water quality may affect the cooking, the amount of heat applied can affect the cooking, the timing of cooking may, the type of utensil used in cooking process may affect the cooking quality. So we use the simple physical method of seeing though how many seeds are hard, they do not absorb water in the absorption process. So what we did is that we were able to develop this low cost test for kind of delineating easy to cook lines and which are hard to cook lines. So another issue that we try to see that if beans are being called as nearly perfect foods, or some people call them as poor means man's meat, because of being very cheap source of plant proteins. So we try to see that what is the nutritional profile of our beans and what are the genetic mechanisms behind them, what are the genes related to these traits. So we did massive studies for kind of evaluation of our material for various micro and macronutrients, various metabolites, primary and secondary, and tried to see what are the genes behind those traits. And we were able to identify these QTLs associated with different nutritional elements in beans. We have already published it. Then we also looked for different, micro and macronutrients like magnesium, potassium, copper, iron, zinc, and we were able to identify low and candidate lines and high candidate lines for developing mapping populations and from trying to see what is the genetic mechanism behind these traits. Apart from doing what we do in the laboratories and the fields, we also try to connect ourselves with the final stakeholders, that is the farmers. So because what happens in plant breeding, breeders try to develop their varieties in their fields using their perceptions, using their mindsets and what suits their kind of working conditions. But sometimes those varieties don't get adapted to farmers' fields, and farmers do not prefer those varieties, and eventually they go out of cultivation. So what we try to do is that more often than none, we try to go to the farmers and tell them what are their challenges, what are their possible opportunities, and what are their perceptions about how varieties should be like. So based on those, we also involve them in varietal selection in a participatory mode so that they can help us to identify better varieties for their farming systems. We also do training of students because there's a need to popularize science and popularize the crops among students, because agriculture, if agriculture goes wrong, nothing else can go right. This is our belief. And so we have to have a kind of understanding and appreciation in our young minds that agriculture is key to our survival, no matter how big our industrialization and other developments are. Ultimately it is agriculture which is going to feed us and keep us fit for doing our job elsewhere. And that keeps us helping students build their interest, build their curiosity, and makes them nearer to science, and they try to see that how they can find a career in science later in their studies. So I see myself in 2013 when I started my postdoc career at Kansas State University. And then from that point onwards, my job has not been doing research only, but mentoring young people, students, boys and girls, and to be able to learn what I am able to do and what I'm able to translate to their minds. And then they could go out and spread this scientific advancement and add to what we currently know. I had an opportunity to train students at my place also, as well as in Kansas State University. And I'm very happy that many of those students are now better placed, doing great job at their places. And some of my students right now, they are in different parts of the world, in US, in Canada, in Italy, Padua. They are doing masters, PhD, they are doing postdocs, and different places, acting as my ambassadors and ambassadors of our state, ambassadors of our country, ambassadors of our institution, and bringing laurels to us. And I'm very proud that they are not only doing their job there, but they are kind of getting recognized for their commitment, their hard work, and the results they bring into their science. I'm very thankful to my honorable vice chancellor, who has probably given me the freedom to do what I do. I'm grateful to my mentor, Professor P.V. Varaprasad, who is one of the world's leading ecophysiologists as far as climate change is concerned, one of the noted figures, who is distinguished professor at Kansas State University, and he's also director of Feed the Future Lab Sustainable Intensification Program that helps farmers, smallholder farmers in Africa and other countries, poor countries, to transform their farming systems into profitable ones. And I'm also thankful to my collaborator, Sajad, who is a plant biotechnologist and expert in genomics, proteomics, and other omics techniques, and who helps us, kind of drive our science in many different and diverse areas. I am very thankful to my institution, to Global Bean Project, to have identified me, as a person who could talk to you for some time about what we do at our places, to my university, to Indian Council of Agriculture Research for funding our research, to United States Department of Agriculture, who gave me an opportunity for postdoc in Kansas, Department of Biotechnology, the state government, and the National Mission on Himalayan, studies, who have helped us, carry forward our research, and also to those young, beautiful boys and girls, our students, who help us create those good data sets through which we are able to progress our science. So I think I have been on time. Thank you very much for listening to me. And if there are any questions that I can answer, I'll be happy to do that. If I'm not able to do that, I'll get back to my place and look for what actually needs to be kind of you know done. Thank you very much. Thank you all.
Thank you. That was amazing. I feel like I speak for everyone. That was so informative. And I'm gonna let it open for questions now if everyone wants to ask or say something about the presentation.
Could I could I start, Gabby? Yeah?
Sorry, I'm muted. Go ahead.
Yeah, that was very interesting. By the way, I work with the IPK closely on various things, but including the breeding of Phaseolus beans. I'm wondering what exactly you talked about, the participatory side of it and farmer interaction. But what actually is the connection between your work and the farmers with respect to the breeding step? So
Yeah. So maybe I don't know whether you have any knowledge about there is one doctor, this probably I don't know whether he's alive. He's from University of Bangor, a great proponent of participatory plant breeding.
Yeah.
I know he came to our place in 2013, and he told us that it would be fruitful to engage farmers in your selection process, even though farmers could not be kind of taken on board in mainstream plant breeding. But he said that initially you can start with participatory varietal selection rather than participatory plant breeding. In participatory plant breeding, farmers are partners in developing material, evaluating, segregating generations, and then finding the final line that could be taken forward. So what we started with is that we used a mother-baby approach for that purpose. So in the mother trials, we would have a large number of genotypes, less number of locations, and the farmers would be evaluating the material based on their preferences and choices. We were amazed to see how it makes a difference. In rice, if you look at the new plant type rice, they say that the leaves should be very erect so that there is no shading of the bottom leaves. They will contribute to photosynthesis.
Yeah.
But when those breeders went to Africa and they told them what kind of rice you like, they said we should have a rice, in terms of plant, we should be drooping. So that when there is drooping leaf, it causes shade and doesn't allow weeds to come because they don't have money for weeding. Number one, an interesting question from farmers. They said, could you give us rice that doesn't make us hungry once we have it in morning or at lunch? We should not feel hungry because we don't have too much rice. So this is something which breeder would never expect or keep in mind where developing a variety. So we will simply look at the plant architecture, the yield, the resistance. These, the socially, adapted issues we never try to understand because we are trained in breeding. We are not trained in sociology, understanding rural, farmers' perceptions. So what we did is that we used this mother approach, having a lot of lines, advanced lines, give it to farmers. And if some of the lines could match their preferences as per their rating, and we would take them in the baby approach. In the baby approach, the number of lines would be less, number of locations would be more, and in that case, we would identify, is there any variety which is adaptable to a larger area of land? So that helped us to develop varieties in rice, in maize, as well as in beans.
Could I just on the question of when you talk about varieties, new varieties, are
Yes.
they being produced and delivered to the farmers by you, bred by you, or are they being bred by what we would might call in Europe breeders?
Yeah, varieties are bred by farmers. Once we breed a variety, then we have a system in India that it goes first in the national testing. In the national testing, they see whether it is fit for release at national level. If it is
Yeah,
not fit for national level, then it goes to the state testing.
yeah,
In the state, it is being tested on the farmer's field, not by us, but by a third party Department of Agriculture. They will evaluate the lines, and based on the data that they give, based on the data that comes from the national testing, a variety is being proposed for release, whether it is better than the existing check or not. If it is better on some parameters like yield, like resistance, or some other traits, then it is being accepted for release. Then we produce
okay,
the basic seed, and then the Department of Agriculture takes this seed forward and produces the foundation and certified seed.
okay, yeah. And the original lines, the crossing that generates the lines is being done by you.
Exactly, sir.
Oh, okay. That was really my question.
Exactly.
Yeah.
Because you see the difference in India and like China. In India, why hybrid rice has not picked up? Because farmers, the product problem is the production of seed. Because in hybrids, you need seed everywhere year.
Yeah,
In
yeah.
China, 50% of the seed is produced by the farmers themselves. So that's why hybrid rice has been so popular in case of China. So breeding, when you put farmer to breeding, making emasculation, doing a cross, I don't think they will kind of fit into that system.
Oh, no, no, no. Thank you.
Thank you, sir.
Thank you, Professor Sofi. If I can much appreciate it, I could not follow the whole presentation. I have no background in biology, but so excuse my question coming up. First of all, I would like to ask you about the elevation. What is the approximate elevation that they grow beans, and how many harvests do you have a year?
Yeah, I told you that we are a mountain ecology.
Sure.
We have the altitude is from somewhere around 1500 meters above sea level to somewhere around 2500 meters above sea level.
Wow.
So in case of those mountainous areas, you cannot expect more than one crop because growing period is very short. It starts
Sure.
somewhere in the month of May and it ends in September because we
Exactly.
have 6 month winters
Sure.
where we have huge snowfall. But
Yeah.
in plains where you could sow early in the March, and because of climate change, our growing period has got extended in the plains. I mean, where the days are a little bit hotter towards October also. So you can have two crops, but for that you need short duration crops, which are bush types and early maturing lines. You cannot have a pole type late maturing line for those double cropping systems.
Indeed. Well, I know the area a tiny little bit, not Kashmir, but well, not Nepali area. So that's why this is where the questions were coming from. And actually, my second question is, when you start, I mean, I understood that you came up with 5 varieties. Is that correct? I mean, that was not your conclusion, but I thought that you said that you actually have 5 bean varieties that you're recommending. Is that correct?
Yes, yes.
All right. So when you were coming up with them, have you based your research on the heirloom seeds, on the seeds that have already been grown in that area before you came on the scene? Sorry, you might have said that, but I have not
Yeah,
understood that.
yeah.
Okay.
In fact, I told in the middle of my, this presentation that despite having varieties from developed from our institution, despite having varieties coming from the national breeding system, still farmers are obsessed with their land races for two reasons. One, those land races are, fairly adapted to the climate change conditions. Number one, they are a part of their culture. Even if they get lower yields with their land races, they will still prefer those compared to the high yielding varieties. Because it is something like they feel that if the blood of their crops is getting contaminated if they use modern varieties. This is perception in those hilly areas where farmers use those age-old traditional varieties.
Mm-hmm. So,
Let me say one more thing in this. So in our breeding program we have been focusing on developing short duration and bush type varieties. But if you look at the farming system in Kashmir, most of the area is in mountains where they grow maize and beans together. So bush types cannot grow together there in the intercropping. You need to have a pole type variety which can grow along with maize and then produce pods.
indeed. Thank you so much. So I could conclude that actually your new varieties, those 5 things that you came up with, they are genetically based on something that you have already found there established before.
Exactly.
Thank you so much.
Exactly.
Thank you. Thank you.
Do we have more questions? We're good. Well, thank you again, Professor Sofi. That was great. I'm glad we have it recorded so everyone can watch it again. And for the first time, for those who weren't here. Thank you so much.
Thank you. It was an honor for me to be, kind of given this opportunity. I don't know how many people know about Kashmir. So I am also a kind of representing my place, my country, my state, and my people. If any of you have ever chance to be here, you would love to be here. This is really a paradise on earth. Wonderful place to be at. Other than crops and research, it's a wonderful place.
Thank you. If anyone wants
Thank
to
you.
say anything or else go to our prep meeting and you can, whoever is not participating can go. And also, Professor, I don't know if you saw in the chat, there is a lot of people thanking you and commenting on your presentation.
Thank you. Thank you. God bless them all and God bless you.
Also, just tell you some news. By the end of this week, I'm gonna send you a newsletter in which you're gonna have also the video of this presentation. And we're gonna have a public meeting in Bean Policy on September 2nd. Okay, so these are the news I have for you. And that's it. Bye-bye.